Hey guys! Yesterday we talked about the history of DNA, and today we're moving on to the structure. So what does DNA look like? And if you guys remember from all the way back at the beginning of the year, we talked about our 4 biomolecules: proteins, carbohydrates, lipids, and nucleic acids. So now we're focusing on the nucleic acids. Remember that these are the chemicals that store the code of life, and they're found in all cells. All living things have nucleic acids. And the examples that we had were DNA and RNA. Well, all nucleotides, their smallest unit, the smallest part of them, is called a nucleotide. Their monomer is a nucleotide. And these nucleotides are made up of a phosphate group, a sugar, and a nitrogen base, as you can see down here. Phosphate group, just like an ATP, same type of phosphate group, the sugar, and the nitrogen base. There's 5 different nitrogen bases: adenine, which is abbreviated to A, guanine, which is G, thymine, T, cytosine, C, and uracil, which is U. These are further broken down into 2 different groups based on their appearance. If you notice, the purines have 2 different rings, a little pentagon and a hexagon, whereas the pyrimidines only have a hexagon shape, only 1 ring. So our purines are A and G, and the pyrimidines are T, U, and C. So our first nucleic acid is DNA, and DNA stands for deoxyribonucleic acid, DNA. And it's named because of its sugar, deoxyribose. DNA is found inside the nucleus or the nucleoid region of all living cells. Now, if you remember, eukaryotes have a nucleus, whereas prokaryotes don't. So the DNA in prokaryotes has to stay in a nucleoid region, a dense area where the nucleoid is all the DNA is all packed up. DNA is double-stranded, a double-stranded helix. What this means is, imagine a ladder that you twist, and that's what DNA looks like. Its 4 bases are A, T, C, and G. So let's look at the structure of DNA in a little more depth here. So you have a nucleotide of DNA right here, and you have repeating nucleotides that make up your entire structure. This is your first strand of DNA, and this is your second strand. And they pair up by having the bases bond with each other. The bases bond according to Chargaff's rule, if you remember correctly. Adenine always binds with thymine, and cytosine always bonds with guanine. The sides, or the backbone of DNA, is made up of 2 different things, a sugar and a phosphate group, and these alternate back and forth to make a very strong backbone for DNA. If you also notice, there's something over here that says 5' end, 3' end. What's that all about? Well, that's based on the sugar. There's 5 carbons in the deoxyribose sugar, and they each have a name, 1' all the way up to 5'. Well, this end of the nucleotide is the 5' end. This bottom part is the 3' end. So DNA has a direction, 5' up here, 3' down here. However, the other strand is called anti-parallel. It goes the opposite direction. So here's its 5' end, and there is its 3' end. So they pair up with each other, but they're going in opposite directions. A little confusing, but you don't really need to care about that too much until we get into replication, where it copies itself, and how it turns into a protein, transcription and translation. So here are your quick questions again. Remember, you can pause the video, answer the questions, and then when you're ready to start it up again, just hit play. So number 1, what are the 3 main parts of a nucleotide? Number 2, what are the 4 bases of DNA, and what are their abbreviations? How do you shorten them? And number 3, describe the shape and the structure of DNA. Our next nucleic acid is RNA. That stands for ribonucleic acid, because of its sugar, ribose. So if deoxyribose is named because of the sugar deoxyribose nucleic acid, is named because of the deoxyribose sugar, we can think that ribonucleic acid is named because of its ribose sugar. It's also found inside the nucleus and nucleoid region of all cells, but RNA can also be found in the cytoplasm and in the ribosomes. Remember, the ribosomes were like the protein factories of the cell, and the cytoplasm was the jelly-like fluid. Unlike DNA, RNA only has one strand, so it's called a single-stranded helix, and its 4 bases are A, U, C, and G. So instead of a T, RNA has a U. So let's look at the structure of RNA a little more closely. Once again, we see the single helix, the single backbone made up of sugars and phosphate groups still, because it's still made up of nucleotides. And it has all the bases in the same way that DNA does, but they're not connected to other bases. That's why it's only single-stranded. It doesn't show up on this picture, but RNA also has a 5' and 3' end. It doesn't have the opposite, because it's only single-stranded, but it does have directionality. Some more quick questions. Number 1, what is the sugar in RNA? Number 2, what is the shape of RNA? Number 3, what are the 4 bases of RNA and what are their abbreviations? And number 4, what are the 3 differences between DNA and RNA? So let's review the 2 structures of DNA and RNA. Both are made up of nucleotides that bond together, and nucleotides are made up of 3 parts: a phosphate group, a sugar, and a base. The difference in nucleotide between DNA and RNA is what type of sugar they have. DNA has deoxyribose, RNA has ribose. They each have 4 bases: T, A, C, and G for DNA, U, A, C, and G for RNA. So the T and the U just switch out. And then their overall shape is a little different as well. Remember, DNA is the double-stranded helix, so it has 2 strands of DNA that run in opposite directions of each other. RNA is a single-stranded helix, so it only has 1 strand. And here's another picture for you to help you remember the shape of those nitrogenous bases. Remember, our purines have 2 rings, and our pyrimidines only have 1. So, number 1, who discovered the structure of DNA? You have to dig deep for that one, remember from last class. Number 2, what is the difference between purines and pyrimidines? And number 3, which bases are purines and which bases are pyrimidines? Also remember from last class we talked about chromosomes. So your DNA likes to organize itself in chromosomes because there's 6 feet of DNA in every single one of your cells. So it has to condense itself in order to fit in there. Once again, you have the double-stranded helix of DNA wrapping around histone proteins, and that keeps winding until you get that nice X chromosome shape. You're going to have to remember that because later on in our next unit, we're going to talk about how those chromosomes get passed on. So our final questions. Number 1, every organism has 1 pair of every chromosome. Where do they get each chromosome from? Number 2, what is the protein found in chromosomes? And number 3, how does 6 feet of DNA fit inside a small cell nucleus? And that's it for the structure of DNA and RNA today. If you guys have any questions, you can post them on the comments or in the discussion board on our Blackboard page. Have a good night.